go. Okay. Hello, class. We're going to start off today with question 1, lesson 8, with the group problems. And this is dealing with an individual that is heterozygous for the following inversion. And so we're given the normal chromosome and then the inversion, or the inverted chromosome. And it says, suppose that these two chromosomes replicate prior to meiosis. And so they produce 4 total sister chromatids. The 2 replicated chromosomes then attempt to synapse during prophase 1. So what is the allele and centromere composition of the 4 products of meiosis if there is a crossover between S and T? So for this question, you kind of draw both of your chromosomes, and it's going to be kind of confusing to explain this, but this one here is the normal chromosome. And so you just draw all the letters on top. And then this one on the bottom is the inverted one. And so what you do for this is you draw it until the inversion takes place. In this case, it's at Q. And then you go to where the inversion is, and it goes all the way from Q to V, yeah, over here. And then wrap it back around this way and go this way. And then you match up all the letters, but you know that the chromosome follows this pattern, if that makes sense. And then, so that would give you, and then to find your 4 products, so you know that 2 of them are going to be the parent, the normal chromosome and the inverted chromosome. And then you know that 3 and 4, they're going to be a little bit wacky. So you're going to start off with this side, and you'll go L, M, N, and then there's the centromere, then O, P, Q, R, and then this is where you kind of switch over to this chromosome. And since T and S had an inversion, you go down here to S, T, U, V, Q, P, O, centromere, N, M, L. And so there's 2 centromeres in that one. And then number 4, you start at the other side, but down on this chromosome, and you go X, W, R, S, T, U, V, W, T, U, V, and then WX down here. So yeah, those, and since there was a crossover between those 2, like they switched places. So yeah, that's question 1. Over to you, Haley.
All right, so number 2, pretty straightforward. All the answers were in the PowerPoints in the notes. So 2A, non-disjunction in meiosis 2 produces 50% aneuploid gametes, and non-disjunction in meiosis 1 produces a mosaic phenotype. For B, the answer is somebody who suffered a paracentric crossover produces both dicentric and acentric chromosomes. And Turner syndrome is an example of a disease caused by monosomy, where familial Down syndrome is caused by Robertsonian translocation, and human liver cells, 4N, 8N, or 16N. A terminal deletion in chromosome 5 causes Cri-du-Chat syndrome, while an inversion of the X chromosome causes type A hemophilia. And for F, a duplication is a change in a chromosome structure that can potentially be beneficial, because losing one's definitely not going to help us at all. When a translocation cross is formed, the alternate segregation pattern is the only one that produces viable offspring most of the time. All right, so moving on to number 3. How many total chromosomes would be found in the humans that have the following conditions? For Turner syndrome, it's going to be 45 because they lost one. Jacob syndrome, it's going to be 47 because they gained one. Cri-du-Chat is going to be 45. Down syndrome is 47. And then type A hemophilia, I'm pretty sure it's just 46 because it doesn't really affect the number of chromosomes that you have. Just for B, which of the terms is not associated with the CMT disease type IA? So the ones that are not associated are all of the options except for diploid and non-disjunction. Because it's going to have trisomy and it's going to be triploid, which also makes it aneuploid. And it goes through reciprocal translocation and duplication. So for C, which are associated with Klinefelters, aneuploid and trisomy were the only ones that I found in the notes. And for D, it was kind of a trick question because type A hemophilia is an inversion. So we said none of them are going to be associated with type A hemophilia. And that's number 3.
All right. And question number 4. So this is a reciprocal translocation question. So this one's kind of confusing to explain too, but I'll do my best. So it says that there's one pair of homologous chromosomes in a mouse that has the following sequence of alleles, and it gives us the sequence. And another pair of the homologous chromosome has this other sequence. Then a reciprocal translocation occurs between one chromosome and each pair, producing the following allele arrangement on 2 of the 4 chromosomes. So this right here is the normal one. These are the 2 normal chromosomes. And keep in mind that these are 2 separate chromosomes. They're not homologous, and so these are 2 separate different ones. But a reciprocal translocation is one that takes place between 2 non-homologous chromosomes. And so the reciprocal translocation takes place in these parts of the chromosome. I don't know if you can see. They essentially swap places. And then the centromere is indicated by this little dot right there. So we're supposed to diagram the synapsis of these chromosomes in prophase 1, and I drew it out. So let me show you the picture. Again, I hope that I can explain this simply, but this right here, I don't know if you can see it. Okay, this right here is diagramming the chromosomes, the synapsis of these chromosomes in prophase 1. So we have our normal chromosomes right here. D, E, F, G, H, I, J, K, L. And then our other normal chromosome is right here, T, S, R, Q, wait, no, sorry. It's on this, it's right here. It's M, N, O, P, Q, R, S, T. So these 2 across from each other are normal chromosomes. And these are our reciprocal translocation ones. So our normal ones will line up like this because the ones that they're lining up with are strange looking, like here, in order to all be connected to the right different alleles on these chromosomes. And so they'll kind of line up like this, matching with each other, like right here, P, Q, R, S, T, D, E, F, D, F, G, H, and yeah, so on and so forth, M, N, O. And so this is how they line up right here. The allele composition of the gametes produced by alternate segregation. So this is the only one that can produce viable gametes. So for this one, what you do, I'm going to try to explain this as clear as I can. This one, what happens is they separate this way. So the 2 normal ones will separate, and then this way, and then these 2 normal ones will separate this way. So, or these 2 inverted ones. So these 2, when they separate and go off with each other, the gametes that they produce are going to be inviable, or they're going to be kind of the messed up ones. But since these 2 go off with each other, they're going to produce viable gametes. And so you have the 2 normal gametes, or the 2 normal chromosomes, and each gamete will have these 2 in them, so they'll be normal. And then 2 other gametes will have these messed up ones, and so they won't be right. Then with adjacent 1, this one deals with, they separate across this way, so horizontally. So these 2 bottom ones will go off and be with each other, and these 2 top ones will go off and be with each other. And when they produce gametes, each one of them will have a normal one with them, but each one of those gametes will also have a messed up one with them. And so all of the gametes are going to be inviable because they're going to have a messed up chromosome. And then adjacent number 2, this one's similar to adjacent number 1, except they separate to the side. So these two go off with each other and these two go off with each other. And the gametes that they produce, kind of like an adjacent one, are all going to be inviable because they're all going to have a messed up chromosome in the gametes with them, as you can kind of see here. So sorry if that's confusing, but that's the best way that I can try to explain how that happens. So that's question number 4.
All right, sounds good to me. So number 5, this one was similar to one of the questions that we had in the practice problems this week. So 5A, a triploid cat is going to have 57 chromosomes because we're going to take 38, which is the diploid number, and divide it by 2 to find n. And then with triploid, we multiply n by 3, and then we get 57. So a cat that is monosomic for chromosome 12 is going to have 37 because it's just going to be one less. And for C, a terminal deletion on chromosome 7, we're pretty sure it's still going to be 38 because it produces a partial chromosome, but it still exists. So for D, a Robertsonian translocation is still going to give us 38 because it doesn't affect the number of chromosomes, it just affects how they connect to each other. A cat that has suffered a reciprocal translocation is going to have 38 still as well. And that's about it.
Perfect. Okay, well that concludes our video for today. So hopefully some of those things helped. And again, sorry if the processes were kind of confusing to explain, but that's what we got for you. Thanks, guys. Stop broadcast.